So the next speaker is our president, Dr. Carole Nicco. So to introduce you, Carole, briefly, Carole, she started her career at University of Paris, Institut Cochin. Then she moved to private company, and then she come back to Cochin Institute, where she worked for many years in many fields, and she conducted many clinical studies related to immunology, endometriosis, and many inflammatory related disease. Now she have another vision that, you know, in order to progress research, we need to have a link with industry. So she now have both sides. She have the industrial vision, and she have the academic and the research vision. Thanks, Carole, for coming, and thanks again for your excellent preparation for the meeting.
Thank you, Marvin, for this kind presentation. Good morning. I hope you're enjoying Paris. So a few months ago, I had to choose a title for this presentation, and I didn't want to use the word poison because arsenic is not a poison. It's a really powerful drug to be used with moderation. So I was wondering, and then I saw my husband training martial art because he is a martial art master. And I said to myself, I could say this is a double-edged weapon. Don't you think a double-edged weapon would be better than a single-edged one? He said, yes, double-edged better. So I said, okay, bingo. I will name my speech a double-edged sword for a unique double benefit against immunological disorder. I was at this time at the Cochin Institute working, and this work has been initiated by BioSenic and MedSenic, where I am working now. So inflammation is an essential defense mechanism. It eliminates danger. This danger can be toxic, infectious, or from immune origin. So when an injury occurs, various mediators released by the tissue will increase the vascular permeability, and some chemoattractants will help the immune cells to go to the site. Then the cells will be activated, and you will have dendritic cells that will present the antigen and activate immune cells such as macrophages. Inflammation is a normal phenomenon, and it's triggered by an external or an internal trigger. After you have an activation and a resolution of this inflammation. That is the physiological response. This resolution sometimes doesn't happen, and you have then a chronification of the inflammation. Here, the eosinophile, the resident macrophages, and T cells are key important cells of this chronification. When you have a chronification, what occurs? There is like a wound healing that is exacerbated, and this exacerbated wound healing triggers fibrosis. Fibrosis is the activation of fibroblasts, and this fibroblast will produce matrix protein that will give a hardness to the tissue. And this multisystemic phenomenon can occur in the eye, skin, cardiac, pancreatic, renal, pulmonary organs. And when you have this multisystemic failure, death can occur. Around 10 years ago, we decided to use arsenic trioxide to evaluate its effect on pro-inflammatory disease and pro-fibrotic disease, on graft versus host disease, chronic graft versus host disease, and systemic sclerosis. Arsenic at this time was already used in human with Trizenox. Trizenox is a molecule used since the 2003, again, myeloid lymphoma. So it's used in human. So we decided to try it in vitro and in vivo on mice model, and we obtained really nice results on these two autoimmune diseases 10 years ago. In 2021 and 2022, this molecule were evaluated in 2 clinical trials in lupus and chronic graft-versus-host disease, and we obtained safety and efficacy really interesting. So how does it work, this arsenic trioxide? This molecule is, this molecule is a metalloid found in soil and water, and it can be a toxic at high level, high dosage. It can enter into the cells. It can enter into the cells through the aquaporin 9, such as hydrogen peroxide. And then in the cells, as you can see, it acts on many pathways, signaling pathways, metabolic pathways, but they are all related, most of them related to reactive oxygen species. So what we decided to do recently with BioSenic and MedSenic is to improve the effect of this arsenic to be able to decrease the dosage in patients. So we decided to associate this arsenic trioxide with a phenton or a phenton-like molecule. First, in vitro, we evaluated arsenic trioxide on HL60 cells, promyelocytic cells, and we used this arsenic trioxide at different dosage from 0 to 5 micromolar. And we evaluated the survival, GSH, and H2O2. Well, we evaluated other things, but I show you here the most representative results. So on the survival, what we observed is that at 5 micromolar, we have the best effects. That is to say, less survival, less GSH production, and H2O2 was not significantly modified. So with this result of 5 micromolar best concentration, we decided to use 1 micromolar to evaluate what was the effect of a phenton or phenton-like molecule on this concentration on the cells. And we use a few phenton-like molecules. We use iron, gold, manganese, zinc, and copper. First here, we evaluated on the same cell, HL60, the H2O2 production. And what we see here is that manganese and copper, so I just have to explain you, so the concentration of arsenic was 1 micromolar, and here you have the increase of the iron, of the metal, from 0 to 1 micromolar. So what we saw is that iron didn't improve the effect of arsenic, neither gold, but manganese, copper, and one of the form of zinc we use was improving the effect of the arsenic trioxide on the H2O2 production. Concerning the cell survival, what we observe, so with the same principle, an increase of the concentration of the metal for each condition, we observed that copper was the best metal, the best phenton-like molecule to optimize the cell survival when comparing with arsenic. So we decided to use copper for in vivo experiments. And for these in vivo experiments, we used 2 models, but I'm going to present only one of them, the chronic graft-versus-host disease model. So just to explain you or remind you, I don't know, what is the chronic graft-versus-host disease? Following a hematopoietic stem cell transplantation, when someone has a blood cancer or leukemia, you sometimes have an activation of the donor immunocompetent cell that you graft to treat the patients. And the issue is the death. So a multisystemic disease can occur. First, during the first period, you have a graft versus host disease. And if it doesn't occur at this period, you can have a second period after around 100 days after the graft, where a chronic graft versus host disease occurs, and it mimics an autoimmune disease. So that's why we choose this pathology. And the prophylaxis and therapy of chronic graft versus host disease is corticosteroids, immunosuppressants, monoclonal antibodies, but nothing to really cure. There's palliative treatments. What was nice is that we have a good preclinical mice model, the same as human. You can graft mice with an allograft, as is explained here. So you irradiate mice. So you deplete the immune system of this mice. You graft the cells from a donor, an allogenic donor. Here we try to have a different donor to have the reaction. And it induced a chronic graft versus host disease. For the control, we have a syngeneic graft, and we don't have any reaction when we do this syngeneic graft. So we did 3 types of groups: the irradiation control, so mice without treatment, only irradiated, no immune system for sure. After 12 days, they die. You have the syngeneic group, control group, to prove that you can manage with the graft. So they survive with no problem, no clinical sign. And you have these allogenic mice treated with either PBS, no treatment. We choose to treat them with arsenic trioxide, 2.5 microgram per gram of mice. It's the low dosage. We also evaluated a high dosage. We evaluate copper by itself for treatment and arsenic trioxide low dosage with copper to see if we had the same effect as the high dosage. What we observed, I won't show you everything. We don't have time. I show you the principal and the most representative results. Here on alopecia, when you do a chronic graft versus host disease, the mice lose their hair. So that's a really easy clinical sign to see. Concerning alopecia, what we observe. So here you will have always first here the syngeneic group, the GVHD group, so the sick mice, and then you will have the copper alone treatment, the arsenic low dosage plus copper, and arsenic high dosage. Here you see that syngeneic mice have no alopecia. Chronic graft versus host disease mice have huge alopecia, as you can see here. And our treatment, arsenic associated with copper, so arsenic low dosage with copper, had a huge effect on alopecia, better than the 5 micromolars treatment that was the high dosage. So we were really happy with that, but it's not the most important, the hair. So we, after at the end of the experiments, we studied the skin fibrosis. And what did we observe? So here you have the histology of the skin, of the ears of the mice, and here you have the thickness of these ears. You see that in GVHD group, there is disorganization, and it's a serious collagen stain here, a lot of collagen. The thickness of the ear is high also, so GVHD is here. And what we observed is that these 3 parameters that are representative of fibrosis evaluated by QRT-PCR are also upregulated: alpha SMA, collagen, and IL-13. So what happened? We observed that with our combined treatment, copper arsenic low dosage, we had a nice results of ear thickness that was decreased comparing to the control group. And also the same result, almost the same result for alpha SMA, IL-13, For the high dosage, it was better for collagen results, but anyway, the results was quite good and really significant for our combined treatment. Concerning the lung fibrosis, because it's also a clinical sign of this pathology we observed in our mice, we observed that there is a disorganization and a fibrosis of the lung in air, the histology, that is also observed with the alpha SMA and collagen as previously in the skin. And our treatment, association of copper and arsenic trioxide, had a very good effect, better here in the lung, with the low dosage of arsenic combined to copper. Same observation in the intestine. I won't give you the details, but these markers of the intestine fibros and dysregulation here are improved, really improved by our combined copper and arsenic low dosage. On inflammation, that's interesting because I told you that fibrosis comes from a dysregulation of inflammation. So this parameter was really interesting. So first, the histology. What we observed is that we have here on this staining, you can not really see good here, but there is an infiltrate. This infiltrate is represented here by the marker CD45, marker of inflammation and lymphocytes in the sick mice. In the treated mice, we obtained the same results with a high dosage of arsenic or low dosage of arsenic and copper. We observed that there was a decrease of these stages of the inflammation compared to the sick mice. So we decided to explore by cell sorter, by FACS, what was going on with these cells. What we observed here on these immune cells is that the memory CD4 T cells that are improved in the pathology are less increased when the mice are treated. So memory effector T cells are decreased comparing sick cells. As well, the naive cells, so that is a good sign because naive cells have not been activated and they are here decreased. So that's really interesting. And what we can say here, that is the same case for the naive T cells here. They are up, so they have not been activated when the mice have been treated with arsenic and copper associated. Oops. Here we have also the MHC class 2 expression on B cells and macrophages that it is increased in the mice that are sick and decrease in the mice treated by either the high dosage or the low dosage plus copper. And what is interesting is this ratio between M1 and M2 macrophages. This ratio is really linked to the fibrosis because M1 macrophages are markers of inflammation and M2 are markers of fibrosis. And here you see that it's really improved. Finally, we decided to evaluate the effect of this mixture on the tumor because we didn't want it to be good for the chronic graft-versus-host disease and improve the tumor at the same time. So we decided to try this treatment on a subcutaneous model of tumor. We injected all the type of, we did the same groups, and we evaluated the increase of the tumor volume. And as you can see, there was no effect on the tumor volume. So it's not something to treat the tumor, the subcutaneous tumor, but to treat graft-versus-host disease. So what are the mechanisms? That's what I showed you at the beginning. We had this cell with arsenic trioxide that was impacting mechanism, signaling pathways, metabolism. And here now we added copper. So I had no real clues, and Raphael gave some of what copper is doing really in our cells. But what we found is that there is not an optimization, but a complement action of the two molecules. And this complementation comes from the fact that they don't use the same pathways. Parallel pathways are used, so they can act depending on the cells differently, but almost on the same pathways. We also studied another pathology, and I won't give you the detail here because it would be too long, but systemic sclerosis is also an autoimmune disease that occurs, and it's a rare disease and have almost the same parameters and almost the same activated cells as chronic graft versus host disease. And we observed the same phenomenon. So we published 2 articles about that. This association of a fentanyl-like molecule that improve arsenic trioxide in autoimmune diseases, recently on chronic graft versus host disease, but also on systemic sclerosis. And with that, we obtained a patent. And I wanted to show you that, to tell you that it's important to bring our molecules, the pathway that we find, to patent to be able to have treatment for the patients, because in fact, it's our aim. So what are the perspective? The perspective with this scientific results is, and this patent, is to be able to treat patients. And to be able to treat patients, we would like to have an oral formulation of this combination. And that is another challenge, the beginning of the second challenge. When you have your molecule, you know how it acts. You want to use it to treat. So we'll have to do a clinical trial phase 1-2 with this oral formulation that we will have to find how to manage to obtain this oral formulation with compound that we only used in liquid formulation until now. And then for a further project perspective, we would like to vectorize these molecules. That is to say, to create ATMP. I don't know if you heard this word, but ATMP is really the new word. If you want to be founded for a new treatment, just go to see ATMP, Advanced Therapy Medicinal Products, that combine those molecules with other technologies that help them to go to the target. This advanced therapy medicinal product I would like to evaluate is small vesicles containing our arsenic copper. And finally, I asked 3 days ago to my husband, okay, my double H word for my title, it was good. It really improves the cut. And he said to me, no, I've never said that. It doesn't improve at all. Only if you are a real good specialist and you know how to deal with the double-edged word. I say, oh my God, what am I going to do? What am I going to say with this title? And then after a while, I said to myself, but it's the same. Arsenic trioxide has to be managed by a professional. You have to be really aware to know what you're doing to use it as a double-edged sword. Thank you. Oh, I would like to thank all the team of the lab I was in last year and for 20 years at the Institut Cochin. And I would like to thank BioSenic and BioSenic, who initiated this research with their arsenic molecule and funded the research. And at this time, I was not in this company. Okay.
Thank you very much, Carole. Thank you.